These are the scientific method notes for Chapter 1 of Environmental Science. We're going over Lesson 2 and Lesson 3. When you start these notes, you want to make sure that you have a piece of paper and a pen handy, so press pause if you don't have those things out right now. Otherwise, you want to open up your folder to the table of contents page, put the date of whatever day you're looking at this, and the title of the notes is Scientific Method. The grade column is going to be left blank because that's for me when I go to grade your notes. When you finish this, you want to make sure that the notes you've written down today are in your folder. They should look like this for your table of contents. You want to make sure they're in your folder because I am going to grade them every so often, and when I do, I will put your grade in that column. These folders you want to make sure do end up back in the classroom, but you can take them as you need. I will give you a warning when I go to grade them. Alright, we're about to start the notes, so make sure that you have a piece of paper and a pen out. Now, when we're doing the notes, you want to make sure that you have a pen, piece of paper, and highlighter. When you go to write things down, the things that are in red are what you're going to write down, otherwise it's information on the board for you. Now, I'm not going to go through and sit here and talk and read the slide to you because you're currently looking at the slide, so read it. That'd be good. So when we're looking at the process of science, a couple of key things that you want to make sure you're aware of is that science can change at any given moment. And that's what's fun about it, is that there's no definite answer. It's about research, it's about the observations, and it's about using the technology, because a lot of our science changes as we get more technology. Alright, so this is the first slide where you're going to write stuff down. So the 3 bullet points on observation, you want to make sure you write down on your paper. What I ended up doing on my paper was I wrote a big bullet point for observations, and then I wrote the 3 topics that observations can be made with the eye, instruments, or by reading scientific material. That observations can happen unexpectedly, or they can be planned, and that a lot of observations lead to questions, and many can be shared with colleagues. So you want to make sure that this is your big bullet point, and that you have these 3 topics on your paper. If you need to pause this because you have not finished writing this down yet, then please make sure that you pause the video at this time. Okay, moving on. The next one is hypothesis. So you want to make sure that you write down hypotheses on your paper, and then you're going to have 4 bullet points underneath that. So with a hypothesis, it's not just a guess, an educated guess. It's about actually looking and observing around you and getting an idea, a testable idea of what you're going to be studying. So a hypothesis can explain a phenomenon. It can also answer a specific question, but you're only required to write down explain a phenomenon. It can generate predictions, so you can use your observations to help create a model. It can help other observations and help point you toward what experiment you want to do. Observations can be supported or rejected by data, that's what that word is over there, and they may start a new hypothesis. You might change it in the middle, and that's okay. You can change your hypothesis as you're going through the experiment. Alright, if you have not finished writing this down, then please make sure you pause the video at this time. The next part is data. This is a big section, and as data should be in any experiment or any report. When you have data, there are going to be a couple of really important vocab words that you should already know. Like seriously, you should already know these. If you don't, then you need to make sure that you memorize these for tomorrow. So there's the independent variable. The independent variable is the variable you want to change. It's the one that you're going to change because it's the results you want to see. So for example, if you're testing if bars of soap lather really well, what you're going to change is the different types of soap that you use, because that's what you want to test. You want to see which soap is better. That would be your independent variable. Your dependent variable is what's going to change, the lathering, whether it lathers a lot or a little. So I'm going to control whether I use Dial or Dove. That's my independent variable, is what type of soap. My dependent variable is how much those soaps lather, based on what I do. Another example of that would be how tall flowers grow. So if I'm going to choose a rose bush, but I'm going to change the amount of water that they get. So I'm looking at how much they grow, that's the dependent variable, based on how much water I give them. That's the independent variable because I'm controlling the amount of water. Lastly, all experiments should be controlled so that you only change one variable. If you are going to change more than one variable, then you're not going to know what results change. So, for example, in the flour example that I gave you, if I use 2 different flours and 2 different amounts of water, then you don't know if it was the flours or if it was the actual water that changed. So you have to make sure that you control everything as much as possible. You put the plants in the same type of pot, the same type of soil, in the same sunlight, in the same air, everything like that, so that you know the growth of the flower is only caused by the water. Same thing with the soap. I'm changing the type of soap I use, whether I use Dial or Dove, and I want to see how much they lather. Well, that means that I have to do both, no one else, because everybody has a different skin type, and I have to make sure that I use the same type of water, because hard water won't lather as much as soft water, like people with water softeners. So you have to make sure you control every amount, otherwise the results you get aren't going to be conclusive. You won't know why they changed. All right, next slide. When you're testing the predictions, you need to make sure that you have data for them. You have to have some sort of table or chart. Before you make a table like the one given on the slide, you want to make sure that you actually have a chart you put it into. You want to do a chart first and then formulate your data as part of your results. Now there's 2 different types of data that you need to make sure you know. There's the quantitative data, which means numbers, because the N in quantitative means numbers. And then you want to make sure that you have qualitative data, which is a viewpoint. It's looking. So the L in qualitative is for looking, how you view it. You don't have to have both types of data, but you do have to have one in order for there to be data. So in the soap example, I'm using qualitative data because I'm looking at how much is lathering. In the flour example, I'm using quantitative data because I'm looking at how much they grow, and that's an amount, that's an actual number. Lastly, looking at this slide, it's analysis and feedback. Anytime you do an experiment, you want to make sure that you show it to peers so that it can be viewed and looked at its validity. You want to see if there's any errors you made, that it was a controlled experiment, because good experiments get messy. A good experiment is complicated, has a lot of parts to it, and involves a lot of thought to make sure that every variable is consistent. For building a theory, it's a little bit more than just a hypothesis that's been tested, because a hypothesis is for a very small area of testability, like the soap, but a theory is for a much bigger concept. So theory of evolution, the theory of gravity. You're not just testing how fast an apple falls, you're testing all of gravity. So both a hypothesis and theories have to be testable, but they also have to be ran through multiple experiments in multiple different ways by multiple different people, but all of those ways, all of those peoples, all of those experiments have to have the same result. And that's why there aren't a lot of theories, is because usually there's one tiny variable that messed it up. So for example, there was a man that was testing a physics theory. I don't remember off the top of my head what it was, but he came through this great breakthrough and published it, and other people started to multiply that experiment. Well, they found out that the reason he got those results was because of the time of day and the place that he was doing his experiment. It so happened that the air conditioning in that building kicked on at the time he was doing his experiment, and that changed his sound waves in his experiment. So little variables that sometimes you can't control will play into your experiment, and that's why other people need to be able to test it as well. All right, lastly, in your notes, you need to tell me which experiment is better. So please go ahead and read both experiments. Experiment A about the acids and bases, and experiment B about the flours, and tell me which one you think is better. Then the second question is, how would you fix the other experiment, the experiment that's wrong? So one of them is right and one of them is way wrong. So tell me, how would you fix the experiment that is wrong? So go ahead and pause the movie at this time so that you're able to look at both experiments, read through them, and figure out which one's better, and then figure out how you would fix the experiment that's wrong. For the how would you fix the experiment that's wrong, you can tell me very plain and simply, I would change this. If you put a lot more detail into it, I would change this because of this and this. If you put a lot of detail into it, then I will give you a lot of points. If you don't put a lot of detail into it, then I'm not going to give you a lot of points. These notes in total are worth 30 points, and I'll give away 10 extra credit points for how well you answer this second question on the board. If you have any questions, please make sure that you ask. If you do not understand how an experiment is run, what type of variables you need, and why you only need one variable in an experiment, then please come and see me during advisory. The main point of these notes is to tell you the different parts of the scientific method, how an experiment's run, and to be able to recognize, like the examples posted right now, what experiment is good and what experiment is bad and why. If you have trouble doing this, then you need to make sure that you come and see me because this is the work that you're going to be doing next week.